This video will be brief, just to fill in some gaps. This graph here shows how enzymes are going to lower the activation energy of reactants. And so when it lowers the activation energy of reactants, a greater proportion of molecules, reactant molecules, are actually able to undergo this chemical reaction because of this change of activation energy. We should know now that enzymes act like a lock and key. They have these specific areas in which the substrates fit into the enzyme called active sites, and then it causes a conformational change. These are highly specific, right? Because enzymes, remember, have that primary, secondary, tertiary, quaternary folding, which makes them really specific for their enzyme action. A quick blurb about the naming of enzymes. Most enzymes name end with A's. Some examples are shown on the next slide. Amylase, lactase, synthase, and they oftentimes tell you what they do, sometimes they don't. For example, lactase enzyme digests lactose, which is what individuals who are lactose intolerant are missing. Enzyme activity can be measured. First, you can measure the rate of substance decreasing, or you can measure the rate of substance products increasing. And this is actually something that you might be doing in lab. They can be affected by temperature and pH. Similar to how you have an optimal work environment where you probably do the best or most work, enzymes are the same way. They have an ideal temperature, they have an ideal pH with which they work, which I'll show you in just a second. They're going to be impacted by cofactors and coenzymes. They require it, and so you want enough of them for the amount of enzyme that you have. And then, of course, you need a good amount of enzyme and a good amount of substrate to be able to produce a good amount of that product. On some enzymes, the products can actually impact how functional these enzymes are, and we're going to talk about this too. So this slide shows the optimal temperature range and the optimal pHs for different enzymes. Our body temperature is 37 degrees Celsius, so it does make sense that usually enzymes' optimal range of temperature is around there. That is when the activity of enzymes are at its best. For these different enzymes, they have different pH ranges that they prefer. Pepsin is one enzyme in our stomach, and our stomach, as you may know, is a really acidic place, and so it makes sense that pepsin's optimal pH is really acidic at around a pH of 2. Salivary amylase is in our saliva, so we'd find that in our mouth. And notice that that's kind of more in the neutral range. Trypsin is an enzyme that's produced by our pancreas, but then it gets released into our small intestine. And notice that it is a bit more basic there. This was mentioned in the video, but cofactors are inorganic, and so those include metal ions like calcium, magnesium, copper, and coenzymes are organic, and these are made of vitamins. And both of these are going to be important for the functionality of enzymes. A lot of enzymes are produced in an inactive form. An example would be pepsin that we just spoke about, that's produced in the stomach. And a lot of our digestive enzymes are actually produced this way, because if they were produced if they were activated as they were produced, it would cause self-digestion. where they would digest the cell that they were produced in, right? Because these are going to break down protein. And so what happens is they are produced in an inactive form and then they get activated by something in their environment. And for the example of pepsin, this gets activated by hydrochloric acid that is also released in the stomach. Some can get activated by adding a phosphate group. Others can get activated by a ligand. What a ligand is pretty much just a word for a small molecule that will bind to it. This was also mentioned in the first enzyme video, but what this graph is trying to show us is that basically an enzyme needs enough substrate for it to work most effectively. So say we only had a little amount of substrate, there's only going to be a little bit of product produced. But say we had a lot of substrate, there's going to be more product produced. And if we have the most substrate, there is going to be a plateau where an enzyme cannot go past because there's only so much enzyme and only so much functionality. Some enzymes are able to catalyze their reactions in reversible ways. An example of this would be carbonic anhydrase, and it is what allows for our buffer system in the body where carbonic acid can be produced out of water and carbon dioxide, and it kind of goes in between this equilibrium state. The law of mass action here states that depending on what concentration is higher or lower, it's going to go from high to low concentration. And so it can go back and forth depending on what the concentration of these products are. Metabolic pathways are pathway reactions that require multiple enzymes, and the product of one reaction is actually going to be the substrate of another, and that is going to occur in a stepwise fashion. And each enzyme, as you can see in this, and each product or substrate here, these are called intermediates, but these are required for the final product to be produced. So all of these are required for this initial substrate to be produced into this final product. Metabolic pathways can be as simple as this, straightforward 5-enzyme pathway, or it can branch off and you can have an enzyme pathway like this, where you can possibly have 2 different final products. These 2 different final products can actually impact how much of each product is produced. This is called end product inhibition. And so say this bottom pathway here, the final product, this F prime, is actually going to come and bind to enzyme 3 prime, and it's going to cause a conformational change. That is an example of allosteric inhibition. When it comes and binds to enzyme 3 prime, causes a conformational change, and then enzyme 3 prime is non-functional. And what that does is it's going to decrease the rate of this F prime product, right? It's going to go from high to low. And then what it's going to do is it's going to increase the product of this pathway. And so this pathway becomes more favored because this one was inhibited. Again, this is an example of allosteric inhibition. This is also an example of negative feedback because you have a final product here that was high, and then because of this inhibition, it decreases the amount of this final product. This is interesting to think about defects, genetic defects. And so there's some mutations that can occur in the body where, say, for example, one of the enzymes, one of the intermediates of the enzymatic pathway, metabolic pathway, is knocked out. It is defective. It does not work. And remember, we said that all of these are required for this final product to be produced. Because we don't have a functional enzyme 3, this final product can't be produced. This is a common metabolic pathway where we take phenylalanine, which is an amino acid. It's found in protein, right? And this shows the metabolism of it, the breakdown of it. And another product of this can be melanin. If you can remember from anatomy, melanin is what we get our skin color from. So there is a defect, a genetic defect caused by a mutation that can knock out this first enzyme, enzyme 1, and this causes a disease called PKU, phenylketonuria. This disease is caused by a buildup of phenylpyruvic acid, and when you have a buildup of this in the body, it causes mental retardation. And so individuals who have this enzyme, who have this defective enzyme 1, the way to treat it is to decrease the amount of phenylalanine in the body. And so they have to go on a really low protein diet. That's the only way that they can treat this and they are unable to produce these, to complete these pathways. Another example of this would be albinism. Albinism, the enzyme 4 is not present, and so these individuals cannot produce melanin.